Hey, good morning, everyone. Is there any questions before we get started today?
I have one.
Yeah.
I missed yesterday's presentation. Is that gonna affect today?
No, that should be posted soon too, so you can rewatch that.
All right, thank you so much.
I have a question.
Mhm.
Where will the lectures be posted?
It should be posted on Canvas.
Okay, so it's not posted on Connect?
No, not Connect, on through Canvas.
Okay,
I should have those posted either today or tomorrow.
okay, thank you.
Any other questions? Okay, so I'm just going to give you guys a little background for Lab 1. Okay, so first off, you have weekly lab reports that are due, and these are submitted through Canvas. So there's a lab report PDF that you can download, and just make sure you submit this in the correct lab report submission area, because there's a lot of labs each week, or there's a lab each week. So make sure you're in the right section. And if you decide to print it and handwrite your labs, you need to download an iPhone scanning feature or some sort of scanning app. Okay, and that way you can scan it and send a PDF. There's also going to be lab practicals. Lab practicals are lab exams, and these are based on the work done for the laboratory portion. And so these may include multiple choice, true, false, matching, short answer, and conceptual questions. And here's some examples, just so you know what to expect. So you'll also be doing a case study project. So the class will be broken up into groups of 3 or 4 students. I think we're going to assign you a patient, and then you'll create a brochure or a poster on that patient. And so you'll include the background, the symptoms, the treatments, and preventions for that disease. Okay, and so this is required to pass the class. And then you have virtual labs via Connect. So I saw that some of you guys have started the labs. I would recommend starting those today. Okay, and then lab practical exams cannot be made up. There's no makeups, there's no exceptions. So make sure to check announcements on Canvas and the syllabus for due dates. Again, with cheating and plagiarism, any student caught cheating or plagiarizing will be subject to the Reedley College disciplinary procedures. So no electronics during a lab exam besides the device in which you are taking the exam. And students with diagnosed disabilities should contact the Disabled Students Program and Services, and then just provide me with a copy of the letter you received detailing the accommodations. Okay, so how are the labs going to look? So there's optional Wednesday Zoom if you have questions. So what you'll do each week is you'll review the PowerPoints for each lab or come to the Zoom sections. You'll download the lab report and complete it using Connect textbook or lab PowerPoints, etc., however you want to write on the lab reports. And then you want to complete your VR labs by each Friday, and then complete the lab exams on days that they are scheduled. Okay, so for lab 1, it's just an introductory lab. And so we're going to be talking about lab safety, and then we're going to be talking about the microscope. So when you're in a lab setting, you always want to follow the lab protocol and the directions given by the instructor. Okay, so this is for your safety and for the safety of everyone around you. You also want to wear proper attire. So usually closed-toe shoes, long pants, a lab coat, eye protection, and gloves. So you just want to protect, just cover yourself up. Food and drink is never allowed. And it's very important to dispose of waste properly. So in the labs, we generate a lot of waste. So biological components need to go into biohazard bags. chemicals into liquid waste collection containers, and needles and glass slides into sharp containers. Okay, so what is a microscope? So we can think of the world as two major sections, the macroscopic environment and the microscopic environment. So microscopes are these tools that scientists use to examine the living world because it's very small, right? So we can specifically observe organisms smaller than what the naked eye can observe. And then most of the time we use light microscopes. So that's just a microscope that uses light to illuminate the specimen. And specifically, we use the compound light microscope. So the compound refers to the fact that there are multiple lenses. So there's an ocular eyepiece, and then there's objectives. Okay, and like I said, you're using a light source to illuminate the sample so that you can see it. There are other microscopes that exist. However, the compound light microscope is the most common. So you do need to know the parts of the compound light microscope. So we have an eyepiece, we have an on-off switch, you have a light adjustment knob, you have a coarse adjustment knob for changes, large changes in magnification. You have a fine adjustment knob for very small changes in magnification. You have a stage knob that controls the stage. You have a light source. You have an iris here, an opening. And then you have a couple stage clips to hold your microscope slide on. And then you have the objective lenses and then the objective nose piece. Okay, so if you saw there's three different objective lenses here that you can rotate through. So typically we have a low power lens, which is 4x magnification, a medium power lens, which is 10x magnification, and a high power lens, which is 40x magnification. Okay, so the eyepiece is 10x magnification. So if you're calculating your total magnification, it's simply the objective magnification times the ocular magnification. So at low power, you're at 4 times 10. So you're at 40x. At medium power, you're at 100x magnification. And at high power, you're at 400x. Okay, so you can really zoom in onto your samples. And what's important here is when you first start looking at a sample, is you want to start on low power, get it adjusted, and then start doing the amplification there in the magnification. Okay, so when viewing objects, it is important to keep in mind the size of the specimen you are viewing, because we're using the microscope to view objects that are not visible with the naked eye, the microscope. We use smaller units of measure to indicate their size. So it's important to keep a relative understanding of the actual size of these objects. So some common units that we use are inches, centimeters, and millimeters. And then we have a couple conversions here for you. So 1 inch is 2.54 centimeters, 1 centimeter is 10 millimeters, and 1 millimeter is 1,000 micrometers. Okay, so another concept that you need to know is your field of view. So that's the circle visible through the lens. So the diameter of the field is the length of the field from one edge to the other. So each line here is a millimeter. Okay, so just to give you a little practice, so if you were given 2 millimeters and you wanted to convert that to micrometers, well, you would look down here and you see, okay, well, 1 millimeter is 1,000 micrometers. So if you have 2 millimeters, you have 2,000 micrometers. And conversely, if you're going backwards, if you have 5,000 micrometers and you're converting that to millimeters, you see 1,000 micrometers is equal to a millimeter. So the answer there would be 5. Okay, so that's it for the microscopes. So you'll have a couple activities in the lab report. So one, you'll label your microscope. So there's a cartoon microscope for you to label there. You'll also calculate the total magnification with the given numbers. The second activity is actually viewing an object. So you'll be looking at the letter E. And you have some questions about that. For the third activity, you'll calculate the field of view with the given numbers, answer some questions. And then there's a couple of videos for you to watch. One here is on the depth of field. I think there's some questions on that. And then the couple of the last ones here are actually preparing slides. And so you'll look at a blood smear image and answer some questions about that. And then it looks like you can disregard the last section. Okay, so is there any questions about microscopes? Okay, so you do have a second lab this week. So lab 2. Okay, and the lab 2 is a little more complicated. So lab 2 is about the macromolecules of life. So just to give you an introduction, organisms are made of cells, but what are cells made up of? So cells are made up of macromolecules, okay? So you are what you eat. Your food provides the basic building blocks for your body. So what are in food products? Well, food products contain carbohydrates, proteins, and lipids. Okay, so the major molecules of life are these biological macromolecules, okay, which are huge. They're usually giant molecules that are linked up into chains. So these long chains are called polymers, and they're made up of a single unit that's just repeating. That's called a monomer. Okay, so just so you remember, poly means many, and mono means one. Okay, and then how these monomers are actually linked into polymers is called the dehydration synthesis reaction. Okay, and it's called dehydration synthesis because when these two come together, a water molecule is removed. Okay, so dehydration synthesis is a way that you build polymers. On the other hand, conversely, when the same polymers are broken apart, water is added. So this is called hydrolysis. Okay, so dehydration synthesis is when you're making polymers, and hydrolysis is when you're breaking down the polymers back into monomers.
Excuse me?
Yeah.
I don't know if it's just me, but I only see the Bio5 Lab 2 on my screen.
Oh, I'm sorry. You see it now?
Yeah, we're all good. Thank you.
Okay, yeah, thank you for fixing that. So you see, I'm looking at these reactions here. So dehydration synthesis is you're removing water to create a polymer, and hydrolysis is you're adding water to make monomers. Okay, so carbohydrates are another macromolecule. So these are also polymers made of monosaccharides. They serve as fuel for the human body, and they're composed of carbon, hydrogen, and oxygen. And these can make complex molecules, including starch, like plants, and glycogen in animals and sugars. So starches are long chains of sugars, but starches do not always taste sweet. Table sugar is sucrose. So your body stores excess glucose as glycogen, which consists of long chains of glucose molecules. Okay, so in carbohydrates, we have monosaccharides, disaccharides, and polysaccharides. Okay, so here's an example of some monosaccharides. So we got glucose, fructose, and galactose. And you can see it just consists of one unit, one sugar. These are called simple sugars. Okay, so monosaccharides can be found in fruits, vegetables, honey, and nuts. Disaccharides are found in sugars and milk. Polysaccharides are found in starchy foods like rice, potatoes, corn, and wheat. Okay, and so when we talk about these, we talk about oligosaccharides, which are chains of a few monosaccharides. So one type is a disaccharide, which is formed by joining two monosaccharides. And so as an example there are sucrose and maltose. Okay, so there's different ways to represent these molecules. So here's a straight chain form of glucose. We also can show these as ring structures with the carbon being shown here, or we can draw it as a ring structure with the carbons implied. Okay, so just different ways that you can draw out these molecules. Okay, so here's another example of a disaccharide. So you're taking two monosaccharides and you're combining those through a dehydration synthesis reaction. You see the removal of water there. And then you have your disaccharide, these two molecules linked together here. Okay, so for polysaccharides now, these are longer chains of monosaccharides. And these usually store energy or provide structure. So it's important to remember that the storage polysaccharide in animals is glycogen, which humans mainly store in their liver and their muscle cells. And the storage polysaccharide in plants is starch, okay? And that's kind of shown here. Yeah, so just to reiterate, glycogen is the storage polysaccharide in animals. Granules of glycogen are stored in cells of the liver. And cellulose is a structural polysaccharide found in the cell walls of plants. So here's a micrograph of cellulose fibrils in a plant cell wall. So these are pretty interesting structures to look at. Okay, so carbohydrates can be split up into two groups. We have simple carbohydrates, which are monosaccharides and disaccharides, and our complex carbohydrates, like our polysaccharides. So we have glucose, fructose, galactose for our monosaccharides, maltose, lactose, and sucrose for disaccharides, and then starches, fibers, and glycogen for polysaccharides. Okay, so another macromolecule are proteins. So proteins are the building blocks of the body and are used in nearly all functions and part of the body. So the monomer of proteins are amino acids. And over here is just an example of an amino acid. So it has a central carbon atom, it has a hydrogen atom, it has a carboxyl group, and it has an amino group. Okay, and when we link these amino acids together, we form peptides. So peptides are chains of amino acids that consist of a central carbon atom bound to a hydrogen, an amino group, and a carboxyl group, in addition to a unique side chain. So we don't show the side chains usually. We just write it as an R. But you will learn about the side chains later in your career. Okay, so here's again showing you that structure where we have an amino group, a carboxyl group, and some side chain. And then here's an example of where you would find proteins, chicken and meat and fish. Okay, so proteins are macromolecules formed by amino acids held by peptide chains. Proteins are the building blocks of the body and are used in nearly all functions and part of the body. So proteins can act as hormones, as enzymes, as antibodies. They're used in growth and maintenance, and they can also be used for the transport of molecules. Okay, so just to remind you, amino acids are the monomer, and then we link those together to form the polymer or the protein. Okay, another macromolecule found in cells are lipids. So these are water-insoluble molecules made of carbon, hydrogen, and oxygen. And these store long-term energy, and they protect vital organs. They also form the cell membrane, and they're subdivided into triglycerides, phospholipids, and steroids. So here's an example of a fatty acid. So you can see it's bound to glycerol here, and you link these long chains up. Okay, and then this over here is a triglyceride. So you can see there's 3 chains here coming off this glycerol. And so this triglyceride contains 1 unsaturated fatty acid. So see this double bond right here, and 2 saturated fatty acids. And what's important to note here is that the double bond forms a kink in the structure here, where it's not as straight. Okay, so the nonpolar bonds that form between the carbon and hydrogen atoms of a lipid cause them to be hydrophobic or water repellent molecules, as opposed to hydrophilic, which is water loving. So it's important to remember those two words: hydrophobic, water repellent, and hydrophilic, which is water loving. This is kind of why water and oil don't mix, because water is polar or hydrophilic, and lipids are nonpolar or hydrophobic. Okay, so lipids are, their monomers or building blocks are fatty acids and glycerol, which I just showed you. So there's those three fatty acids and that one glycerol. The function is a structural support for the cell, energy storage, and cell signaling. Like I said, lipids do not dissolve in water, and they're found as fats and oils, phospholipids, and steroids. Okay, so here's an example of lipid found in a cell membrane. So we call this a lipid bilayer. And these are both polar and non-polar. So you have polar heads or hydrophilic heads, and you have non-polar tails or hydrophobic tails. And these create this structure where you have a non-polar interior and then a polar exterior that can interact with water. Okay, the last macromolecule here is nucleic acids. So these are made of monomers called nucleotides that make up the information storing molecules in our cells, DNA and RNA. Okay, and so there's 4 nucleotides that are found to make up nucleic acids. We have adenine, thymine, cytosine, and guanine. We also have uracil, which is found in RNA. That's not shown here. And the function of this is to store and express genetic information. And just to repeat again, the 5 nucleotide bases are adenine, thymine, cytosine, guanine, and uracil. Okay. So there are tests to do to determine what macromolecules are in a sample. So what you'll be doing in the lab is these tests. So you'll be testing for carbohydrates. So to test for carbohydrates, we use Benedict's solution. And in the presence of monosaccharides or carbohydrates, the solution will turn orange. Okay, so for Benedict's solution, you want to remember orange. If no monosaccharides are present, it'll turn blue. Okay, and so here's an experiment down here where you're starting with no sugar and then slowly increasing the amount of sugar. Okay, and you can see here that orange indicates the sugars and blue indicates there are no sugars. For proteins, we use Byret's solution. Okay, so it turns violet when it's positive, shown here. So when there's proteins present and you add the solution, it turns purple or turns this violet color. And then it turns blue if there's no protein present. Okay, for lipids, we use bile salts. So bile salts can be used to test for the presence of lipids in a solution. And if layer of clumps is positive, this means that lipids are present. If there's no layer of clumps, then it's negative, meaning there are no lipids present. So you should note here that there's no color change here. You're just looking for the presence of clumps. So a positive test will show these little clumps, and a negative test will show this clear sample. Okay, so for your lab, you'll be doing an experimental procedure where you're trying to figure out what's in the unknown sample here. And you'll be using Benedict's solution, iodine, Byret's, and bile salts. Okay, so that'll be your lab. Okay, so just to give you a quick review, there's 4 main macromolecules found in cells. We have nucleic acids, carbohydrates, lipids, and proteins. And it's important to know what the monomer is of each of those. So for nucleic acids, we have nucleotides. For carbohydrates, we have monosaccharides. For lipids, we have glycerol and fatty acids. And for proteins, we have amino acids. Okay, and then the functions of each of these. So for nucleic acids, these store and use genetic information. For carbohydrates, this is used for fuel for metabolism. Lipids, they're used for structural support for the cell, energy storage, and cell signaling. For proteins, they assist in a variety of cell functions like enzymes and things like that. Okay, so for your lab, you'll use this information to fill in the table and come up with some examples. And then you have a couple of videos to watch here. One is the testing for monosaccharides. Another is testing for polysaccharides. You'll be testing to see if proteins are present. And then you'll answer some review questions based on those videos. Okay, and that's it. Are there any questions about the labs?
Yeah, where are those videos and review questions posted? Is that on the McGraw?
So that's in the PowerPoint slide in Canvas under the lab section. Here, I can show you. Can you also see my screen?
Yes.
Yeah.
Yeah, so those are right here. So lab 1 overview, read and review. And lab 2 overview, read and review. So those are the PowerPoint slides I just showed you. So those links are in there.
All right, thank you.
Yeah, and then to get to the labs, you'll go through Connect. And then when you finish your lab, you can submit it here. And I'll have to fix the dates here.
I have a question.
Yeah.
If we like totally like bomb the lab, is it gonna affect our grade, or can we redo it?
I think you only get one submission.
Okay.
Any other questions? Yeah, I would recommend doing the labs now. I would just get started on them. And if you have any questions, you can shoot me a message on Canvas or contact me.
For the homework that we have to do, if we go to Canvas under assignments, is the same to do it there as to go to the McGraw book?
No, so you want to
Or
go,
to Connect?
yeah, you want to go through Connect.
So it's better to do all the assignments in Connect.
Yeah, but make sure you go through Canvas to get there. So come into Canvas and then go to Connect,
Okay,
and then it should launch you out of Canvas. And then your labs are down here. And we're doing
Okay.
lab 1 and 2 this week.
and then we also have like some assignments on where it says lecture and all IRAT or something like that. Do I do those in Connect too or on assignments?
Yeah, so you want to do those through modules.
Okay,
And so you have your lab report that you submit here, and then your discussion board that you submit here.
okay.
Any other questions? Yeah, so go ahead and just get started on the labs and let me know if you guys have any questions, okay?
Okay, thank you.
Yeah, thank you. All right, sounds good, everyone. I'll see you later.
Bye.
Okay, bye. Bye.